friends, I am Tehseen Khan, Assistant Professor, Sagar. Dear friends, today we are going to discuss about an interesting and learning episode from one of the important unit of Immunology and Immunological Preparations 1 of subject Pharmaceutical Microbiology, B.Pharm 8th semester. Scripted by Dr. Rupal Dubey, Assistant Professor Guest Faculty, Department of Pharmaceutical Sciences, Dr. Hari Singh Gaur, Central University, Sagar, Madhya Pradesh. The goal of this episode is to elucidate the characteristics of a healthy immune system, focusing on the principles of immunity and the cells that participate in host protection. We also discuss about the innate, adaptive, humoral and cellular immunity. Let's start our episode while taking a glance at what we are going to discuss further. Module 1. Introduction and Principle of Immunology. Module 2. Antigen and Haptens. Module 3. Immune System and Immune Tissues. Module 4. Innate and Adaptive Immunity. And Module 5. Humoral and Cellular Immunity. Dear friends, let's begin our module number 1, Introduction and Principle of Immunology. Introduction. Immunology has its origin in its study of how the body protects itself against infectious diseases caused by microorganisms such as bacteria, viruses, protozoa and fungi, and also parasitic organisms such as helminth worms. Important initial barriers to infection are physical. Example, the skin, enhanced by substances secreted by the body, such as saliva and tears, that contains molecules that can neutralize bacteria. The internal mucosal tissues, example lungs and airways and gut, are coated with mucus that is able to trap potential infectants. In the airways, mobile ciliate hairs work together to transport contaminants away from vulnerable areas. Tissues such as the skin, mucosal surfaces and airways also contain populations of immune cells that can respond to infectants that breach these physical defenses. In its most complex forms, the immune system consists of two branches: the innate immune system that utilizes certain hardwired strategies to provide a rapid general response when alerted by certain typical signals of infection, essentially forming a first line of defense, and the adaptive immune system that is able to develop highly specific responses and a persistent immune memory to target infection with extraordinary accuracy. Both systems work in close cooperation, and to an important extent, the adaptive immune system relies upon the innate immune system to alert it to potential targets and shape its response to them. Principle of Immunology. The immune system, composed of innate and acquired immunity, allows an organism to fight off foreign pathogens. Healthy immunity accomplishes 4 essential principles. Number 1, ability to detect and fight off infection. Second, ability to recognize a host's own cells and self, thereby protecting them from attack. Number 3, a memory from previous foreign infections. And number 4, ability to limit the response after the pathogens has been removed. In an unaltered state, the intricate network of immunologic organs and cells create an environment for proper host defense. Without adequate execution of immunologic mechanisms, a host is rendered defenseless against pathogens. Conversely, an unchecked immune response can be self-destructive. As a result of either of these untoward sequelae, immune dysfunction can elicit diseases state in the host. Moving to the next module, that is module number 2, Antigen and Haptens. Antigen. In immunology, an antigen is a molecule capable of inducing an immune response on the part of the host organisms. Though sometimes antigens can be a part of host itself, In other words, an antigen is any substance that causes an immune system to produce antibodies against it. Each antibody is specifically produced by the immune system to match an antigen after cells in the immune system come into contact with it. This allows a precise identification of the antigen and the initiation of a tailored response. The antibody is said to match the antigen in the sense that it can bind to it thanks to adaptations performed to a region of the antibody. Because of this, many different antibodies can be produced, which is specifically to bind many different antigens while sharing the same basic structures. In most cases, an antibody can only bind one specific antigen. In some instances, however, antibodies may bind more than one antigen. An antigen is a molecule that binds to age-specific receptors but cannot necessarily induce an immune response in the body by itself. Antigens are usually peptides, polysaccharides or lipids. In general, molecules other than peptides, that is saccharides and lipids, qualify as antigen but not as immunogen, since they cannot elicit an immune response on their own. Furthermore, for a peptide to induce an immune response, activation of T cells by antigen-presenting cells, it must be a large enough size, since peptides too small will also not elicit an immune response. The term antigen originally describes a structural molecule that binds specifically to an antibody. It expanded to refer to any molecule or a linear molecular fragment that can be recognized by highly variable antigen receptors, that is B cell receptors or T cell receptors of the adaptive immune system. The antigen may originate from within the body, that is self-antigen, or from the external environment known as non-self. The immune system usually does not react to self-antigens under normal homeostatic conditions due to negative selection of T cells in the thymus and is supposed to identify and attack only non-self invaders from the outside world or modified harmful substances present in the body under distressed conditions. Antigen-presenting cells present antigen in the form of peptides on histocompatibility molecules. The T cells of the adaptive immune systems recognize the antigens. Depending on the antigens and the type of histocompatibility molecules, different types of T cells are activated. For T cells receptor, that is TCR, recognition, the peptide must be processed into small fragments inside the cell and presented by a major histocompatibility complex, that is MHC. The antigen cannot elicit the immune response without the help of an immunologic adjuvant. Similarly, the adjuvant component of vaccines plays an essential role in the activation of the innate immune system. Type of antigens. Antigens can be classified according to their source. Exogenous antigens. Exogenous antigens are antigens that have entered the body from the outside, for example, by inhalation, ingestion or injection. The immune system's response to exogenous antigens is often subclinical by endocytosis or phagocytosis. Exogenous antigens are taken into the antigen-presenting cells, that is APCs, and processed into fragments. APCs then present the fragments to T helper cells, that is CD4+, by the use of class II histocompatibility molecules on their surfaces. Some T cells are specific for the peptide MHC complex. They become activated and start to secrete cytokines, substances that activate cytotoxic T lymphocytes, that is CTL, antibody secreting B cells, macrophages and other particles. Some antigens start out as exogenous and later become endogenous, for example, intracellular viruses. Intracellular antigens can be returned to circulation upon the destruction of the infected cell. Endogenous antigens. Endogenous antigens are generated within normal cells as a result of normal cell metabolism or because of viral or intracellular bacterial infection. The fragments that are then presented on the cell wall surfaces in the complex with MHC class I molecules. If activated, cytotoxic CD8+ T cells recognize them. The T cells secrete various toxins that cause the lysis or apoptosis of the infected cell. In order to keep the cytotoxic cells from killing cells just for presenting self-proteins, the cytotoxic cells, that is self-reactive T cells, are deleted as a result of tolerance, that is negative selection. Endogenous antigens include xenogenic, that is heterologous, autologous and idiotypic or allogenic, that is homologous antigens. Autoantigens. An autoantigen is usually a normal protein or protein complex and sometimes DNA or RNA that is recognized by the immune system of patients suffering from a specific autoimmune disease. These antigens should not be under normal conditions the target of the immune system, but their associated T cells are not deleted and instead attack. Neoantigens. Neoantigens are those that are entirely absent from the normal human genome as compared with non-mutated self-antigens. Neoantigens are of relevance to tumor control as the quality of the T cells pool that is available for these antigens is not affected by central T tolerance. Technology to systematically analyze T cells reactivity against neoantigens became available only recently. Viral antigens. For virus-associated tumors such as cervical cancer and a subset of head and neck cancers. Epitopes derived from viral opening readings frame contribute to the pool of neoantigens. Tumor antigen. Tumor antigens are those antigens that are presented by MHC class I or MHC class II molecules on the surface of tumor cells. Antigens found only on such cells are called tumor-specific antigens, that is TCAs, and generally result from a tumor-specific mutation. More common are antigens that are presented by tumor cells and normal cells called tumor-associated antigens, that is TAAs. Cytotoxic T lymphocytes that recognize these antigens may be able to destroy tumor cells. Tumor antigens can appear on the surface of the tumor in the form of, for example, a mutated receptor, in which case they are recognized by B cells. For human tumors without a viral etiology, novel peptides, that is neoepitopes, are created by tumor-specific DNA alterations. Next is haptens. Haptens are small molecules that elicit an immune response only when attached to a large carrier such as a protein. The carrier may be one that also does not elicit an immune response by itself. Once the body has generated antibodies to a hapten carrier adduct, the small molecule hapten may also be able to bind to the antibody, but it will usually not initiate an immune response. Usually, only the hapten carrier adduct can do this. Sometimes, the small molecule hapten can even block immune response to the hapten carrier adduct by preventing the adduct from binding to the antibody, a process called hapten inhibition. The mechanisms of absence of immune response may vary and involve complex immunological mechanisms, but can include absent or insufficient co-stimulatory signals from antigen-presenting cells. At the molecular level, an antigen can be characterized by its ability to bind to an antibody's variable Fab region. Different antibodies have the potential to discriminate among specific epitopes present on the antigen surface. A hapten is a small molecule that changes the structure of an antigenic epitope. In order to induce an immune response, It needs to be attached to a large carrier molecule such as a protein. Antigens are usually proteins and polysaccharides, and less frequently lipid. This includes parts, that is coats, capsules, cell walls, flagella, fimbriae and toxins of bacteria, viruses and other microorganisms. Lipids and nucleic acids are antigenic only when combined with proteins and polysaccharides. Before moving to our next module, that is about immune system and immune tissue, I would like to suggest you to visit our website www.cec.nic.in for further readings and learning of more topics associated with your B.Pharm syllabus. Here, you will find many lectures, e-contents, FAQs, LORs and many more. Moving to our next module, that is module number 3, Immune System and Immune Tissues. Immune system. In its most complex forms, the immune system consists of two branches, the innate immune system that utilizes certain hardwired strategies to provide a rapid, general response when alerted by certain typical signals of infection, that is essentially forming a first line of defense, and the adaptive immune system that is able to develop highly specific responses and a persistent immune memory to target infection with extraordinary accuracy. Both systems work in close cooperation and, to an important extent, the adaptive immune system relies upon the innate immune system to alert it to potential targets and shape its response to them. Immune tissues. All immune cells originate in the bone marrow, deriving from homeopoietic stem cells. But an important set of immune cells, that is T lymphocytes, undergo maturation in an organ known as thymus. The thymus and bone marrow are known as primary lymphoid tissues. Secondary lymphoid tissues, namely the lymph nodes, spleen and mucosa-associated lymphoid tissues, that is MALT, are important sites for generating adaptive immune responses and contain the lymphocytes, that is key adaptive cells. The lymphatic system is a system of vessels draining fluid derived from blood plasma from body tissues. Lymph nodes that house lymphocytes are positioned along draining lymph vessels and monitor the lymph for signs of infection. MALT tissues are important in mucosal immune responses and reflect the particular importance of the gut and airways in immune defense. The spleen essentially serves as a lymph node for the blood. Module 4. Innate and Adaptive Immunity. Innate immunity. Mast cells and basophils are innate cells type that, when activated, secrete histamine, which can be an important inflammatory mediator produced in response to initial tissues damage as a result of infection. Mast cells are tissue resident, example in mucosal tissues, while basophils are found in the blood. In particular, they play a key role in the so-called allergic responses. Innate immunity comprises both cellular and humoral in solution elements. The cellular elements are represented notably by phagocytes, specifically neutrophils and macrophages, that can respond to signs of infection, that is, inflammation, in the tissues and home in an infective bacteria before neutralizing and engulfing them, that is, phagocytosis. Recognition of microorganisms by the innate system occurs via characteristic pathogen-associated molecular pattern, that is PAMPs, on microbial surfaces, and an important family of innate receptors called pattern recognition receptors, that is PRRs, are responsible for this, notably including toll-like receptor, that is TLRs. The natural killer, that is NK cells, is another important innate cell that is able to detect and target intracellular infection of body cells by viruses. A further specialized innate cell is the eosinophil that plays a particular role in targeting larger infective organisms such as parasitic worms. The complement system represents the humoral arm of innate immunity and consists of a number of proteins found in solution in the blood that can interact directly or indirectly with infective bacteria through different activation pathways. Inflammation as a result of infection allows plasma containing complement proteins to enter infected tissues. Once activated, the member protein assembles to form complexes on the surface of microbes that punch holes in the membrane. The complement activation pathway are termed the classical pathway, the alternative pathway, and the mannose binding lectin pathway. Adaptive immunity. Key to the adaptive immune response is the lymphocyte. There are several subtypes, however, these fall under two broad designations, T lymphocytes and B lymphocytes, commonly known as T cells and B cells. Although both originate in the bone marrow, T cells mature in the thymus while B cells mature in the bone marrow. During an organism's early development, a large number of B and T cells are produced, each of which has the ability to recognize a specific and essentially unique molecular target. An important aspect of this maturation process is that for both of these cells that recognizes targets within the body, that is self tissue, are identified and weeded out. An additional aspect of the maturation process for T cells is that further distinct subsets and produced helper T cells, also called CD4 plus T cells, and cytotoxic T cells, also called CD8 plus T cells. The individual specificity of lymphocyte is key to the generation of adaptive responses. Adaptive immunity utilizes many kinds of receptor to coordinate its activities. T cells carry T cell receptors, that is TCR, while B cells carry B cell receptors, that is BCR, and variations in the fine structure of these receptors account for the individual specificity described above. In addition, another set of receptors encoded by the major histocompatibility complex, that is MHC, play an important role in adaptive immunity, that is MHC class I receptors are displayed on a majority of body cells, while MHC class II receptors are restricted to antigen presenting cells, that is APCs. Both of these receptor types interact with TCRs. The adaptive immune response consists of two branches: a cellular adaptive response effective by cytotoxic T cells and a humoral adaptive response effected by B cells. The former is directed specially towards pathogens that have colonized body cells or body cells that have become malignant, as in cancer. The latter generally targets pathogens or molecules, antigens, that are free in the bloodstream or present at mucosal surfaces. As suggested by its name, the helper T cell plays a central role in both of these responses, since once activated, it can shape the subsequent immune response through the particular molecule that it secretes, in particular controlling the activation of other cell types. As such, it is an important gatekeeper. Two subtypes of helper T cells, Th1 and Th2, have been identified as being responsible for guiding adaptive responses towards either a cellular profile, that is Th1, or a humoral profile, that is Th2. Th17 cells have recently been identified and are thought to play a further specialized role. Effective regulation of immune responses is also vital to ensure that they don't themselves cause unnecessary tissue damage, and regulatory T cells, that is Tregs, are a subset of T cell that play an important role in this process. Moving to our next module, module number 5, humoral and cellular immunity. Humoral and cell-mediated immunity are together responsible for coping with foreign bodies like viruses and bacteria that enter a human organism and cause various diseases. Humoral immunity in the adaptive immune system. Humoral immunity is responsible for viruses and bacteria that have not yet penetrated into the cell. The cells that make humoral immunity possible are known as B cells, which contain B lymphocytes. B lymphocytes originate in bone marrow and are a subset of white blood cells, have around 10,000 proteins in them, also known as membrane-bound antibodies or also immunoglobulins. Each B cells has its own distinct set of antibodies. All these antibodies are produced by the gene of the particular cells, and their combination make up as many as 10 billion variations on the human organism. The humoral immune system in the human organism has developed in such a way that it is able to organize its B cells via bonding in order to fight any potential virus or bacteria that enter the human body. When a B cells find the combination that is able to cope with a virus, it starts to replicate and also differentiate. Some B cells become memory cells, that is, accumulate information about the construction of an antibody. So, in the future, the system will work faster when invaded by the same type of virus. And effector cells, antibody factories producing replicas that make around 2000 cells a second. Specific antibodies bond with viruses and tag them to the system's phagocytes that destroy them, the process known as opsonization. Figure number 2 represents a step-by-step scheme of production of antibodies by B cells, where you can see a B cells is triggered when it encounters its matching antigen. Now the B cells engulfs the antigen and digests it. Then it displays antigen fragments bound to its unique MHC molecule. Now this combination of antigen and MHC attracts the help of a mature matching T cell. Cytokines secreted by the T cells help the B cells to multiply and mature into antibody producing plasma cells. Released into the blood, antibodies lock onto matching antigens. The antigen-antibody complexes are then cleared by the complement cascade or by the liver and spleen. Cellular immunity. In the adaptive immune system, cell-mediated immunity is responsible for viruses and bacteria that have penetrated into the cell. The cells that make cell-mediated immunity possible are known in biology as T cells, which mature in the thymus. There are two types of T cells. Helper T cell work as an alarm in the cell-mediated immune system. When dendritic cells find dangerous cells, they send the information about them to T cells receptor of the helper T cells. Each T cell has its own unique combination of proteins that binds to specific antigen that are presented to it. There are naive T cells that have never been activated by anything. If such a cell binds, it activates and is no longer naive. It starts to produce many copies of itself. Effector T cells. Once effector cells are activated, they start releasing cytokines molecules, which raise the alarm to the other part of the immune system. Essentially, these cytokines work like alarm bells, ordering B cells to activate and divide more often. This process is known as T-dependent activation. Memory T cells accumulate information in order to cope with the future information. They live longer other types of cells. Cytotoxic T cells. These cells attack foreign cells that infiltrate the native human cells, not necessarily cells. from the outside, but also those originating in the human organism. For example, cancer cells. Humoral versus cell-mediated immunity. What is the difference between humoral immunity and cell-mediated immunity? Let's assume a human organism gets infected by a virus, which when it appears in the human system, just floats around in the fluid of the organism. That is the stage when humoral immunity with its response start to work on this virus. On the other hand, the virus has infiltrated inside the cell. It makes it produce other viruses. This is the stage where cell-mediated immunity with its mechanisms starts to work. The two systems of immunity, humoral and cell-mediated, can be viewed as a fail-safe mechanism. If every time a virus provokes antibodies, there is a chance that the immune system can start producing cells that become specific not to foreign objects but to molecules originating in a healthy human organism. This possible random mutation, which potentially causes disease, known in biology and medicine as autoimmune, is the reason for the safe mechanism which is provided by the division to the humoral immune system and the cell-mediated immune system. Here is a comparison chart between humoral immunity and cell-mediated immunity. In humoral immunity works on viruses and bacteria that are outside of the cells, whereas cell-mediated immunity works on viruses and bacteria that have penetrated inside the cells. In humoral immunity, it is a simpler system, while the cell-mediated immunity is a more sophisticated system. Humoral immunity activates B lymphocytes, whereas cell-mediated immunity activates T lymphocytes. Summary. The immune system consists of factors that provide innate and acquired immunity and has evolved to become more specific, complex, efficient and regulated. One of the principal functions of the human immune system is to defend against infecting and other foreign agents by distinguishing self from non-self, that is foreign antigens, and to marshal other protective responses from leukocytes. The immune system, if dysregulated, can react to self-antigens to cause autoimmune diseases or fail to defend against infections. Immunity refers to protection against infection. The immune system is the collection of cells, tissues and molecules that functions to defend us against infectious microbes. The coordinated reaction of the immune system against infections and other foreign substances is known as the immune response. Abnormalities of the immune system that results in defective immune responses make individuals susceptible by infections by viruses, bacteria, fungi and parasites. This antimicrobial defense function of the immune system is essential for our ability to survive in an environment that is teeming with potentially deadly microbes. In this lecture, we helped you to understand the basic features of normal immune responses and immune-mediated diseases. I will return with next lecture where discuss about immunology in much greater depth, when we will discuss how the immune system combats microbes and how its abnormalities cause a variety of diseases.